Okay, so as you'll soon see, this entire week should be a review of things that you have learned in Biology 111. If they are completely new to you, please let me know and we can go back over some of the things that you need to have known from Biology 111. So, in this section, we're going to talk about some of the differences between the two major types of cells. Remember, the two major types of cells are prokaryotic cells and eukaryotic cells. So go ahead and pause the screen here. I'll give you a few seconds. And I want you to tell me what the differences are between these two types of cells. Okay, so if you said that prokaryotic cells do not have a nucleus and the eukaryotic cells do have a nucleus, you were correct. So let's talk a little bit more about these prokaryotic cells. The two main types of prokaryotic cells are going to be bacteria and archaea. For our purposes, we are primarily only going to be talking about bacteria. So there are three main shapes that bacteria can take. And here are some examples of those. So bacteria can be a rod shape, sort of an elongated oval shape. This is known as bacillus. So some of the ones in this category would be things like E. coli or Bacillus anthracis. Do you guys have a guess on what the Bacillus anthracis is the causative agent for? If you said anthrax, you are correct. Another shape would be the coccyx, or circular, spherical shape. Some examples of ones in this category would be Streptococcus pneumoniae and Neisseria meninginitis. Another type would be a spirochete. These would be known as this flexible, sort of wormy-shaped bacterium. Spirillum are the same shape, but they're going to be a little bit more rigid. So members of this category typically are a little bit more exotic. And so two examples would be Borrelia burgdorferi, which is the causative agent for Lyme disease, and Treponema pallidum, which is the causative agent for syphilis. So this is the basic structure of a prokaryotic cell. Again, this is E. coli, assuming that it's this bacillus or rod shape, and you're going to see several characteristic components. So some of them are going to have these pili or fimbriae. The very outermost layer is going to be a capsule. Some may have this, some may not have this. This can be very gooey, very sticky, and it can allow a lot of these bacteria to adhere to many surfaces. And so this is why just simply rinsing your hands isn't often good enough. You need to physically remove them by using things like soap and water. And these are also a big problem in the hospital setting, as you might imagine. If the doctors or nurses don't wash their hands thoroughly enough, that they may be carrying these bacteria from one patient to another patient, and hopefully that doesn't happen. Prokaryotic cells are also going to have a cell wall. We'll talk more about that in a little bit. As with all cells, they're going to have a plasma membrane or cytoplasmic membrane. Again, moving inward, they're going to have cytoplasm, that's the fluid on the inside here. They're going to have ribosomes, that's where we're going to be doing protein synthesis. And then they're going to have their DNA. It's often centered in this sort of a central region, they call it a nucleoid. You may also have, some bacteria may also have a flagella. So looking at the bacterial cell wall, there's two main types of bacteria, gram-positive bacteria and gram-negative bacteria. And the major difference comes from a stain that you can do, and if you've taken microbiology, you should know about this, a gram stain. If it's going to be positive for this stain, it's going to have a structure like this. It's going to have the plasma membrane, and then it's going to have a layer of the cell wall known as peptidoglycan. It may have some other polysaccharides and lipids and things that are coming up out of here. But the major difference is that this peptidoglycan is going to be the outermost layer. So what happens is when you add this gram stain, this peptidoglycan gets stained, and the result is a positive on this particular stain. If something is gram-negative, there's going to be an extra outer membrane layer here, and before you get to this peptidoglycan. And so it's going to be negative for this particular stain. So let's look at some of the differences between eukaryotic cells and prokaryotic cells. they're going to have some components in common. Primarily, they're going to both have a plasma membrane, they're both going to have cytoplasm, every cell is going to have DNA, and they both need ribosomes so that they can do their protein synthesis. However, the eukaryotic cells, as you hopefully remember, are going to be a lot more complicated. So it's probably too small for you to read many of these different words, but I'm just going to go over some of the parts that are important for these animal cells. The next slide after this will be plant cells. So all of these eukaryotic cells are going to have a nucleus that is going to be the command center of the cell. This is going to be where the DNA is located. Remember that encasing this nucleus is going to be the nuclear envelope. It's a double membrane layer and it has these little pores. And they're going to be really important because they're going to be the ones that are going to allow the RNA, specifically the mRNA, to be able to leave the nucleus when it is complete. You will have very condensed regions of DNA, and this is known as the chromatin. And remember, the DNA is always going to be associated with many different proteins. We'll talk more about that in a minute, but they're called histones. You also have a sort of a darkened spot in the nucleus. This is known as the nucleolus, and this is the region that's going to produce the subunits of the ribosomes. So this is probably the most important for this course, but I will just briefly go over the others. The mitochondria, don't forget, those are really important for cellular respiration. That is going to be used for basically taking energy from glucose. The rough ER, or endoplasmic reticulum, is called rough because it's studded with ribosomes. Remember, the role of a ribosome is to synthesize proteins. So proteins that are made at this rough ER, because this is really involved with secretion and so forth, these are often proteins that are destined for secretion from the cell. Ones that are made at the ribosomes out in the cytoplasm, they're usually destined to stay inside the cell. You'll have a smooth ER. This is lax ribosomes, and it's used for synthesizing lipid molecules. You also may do some detoxification reactions here, and so on. The Golgi apparatus is important for processing, packaging, and secreting modified proteins. So proteins, lipids, they're going to come from this ER, either the rough ER or the smooth ER. They're going to come to the Golgi, get modified, and sent on their way. Okay, so plant cells are going to have many of the same parts. Particularly, they're going to have a nucleus, they're going to have ribosomes, they're going to have ER, both types, they're going to have a Golgi and cytoplasm. But a couple of the main differences is that cell wall, plant cells are going to have a cell wall. So this is a very rigid structure on the outer surface, going to help shape, support, and protect cells. They're also going to have this large central vacuole. This is primarily going to be a large fluid-filled sac. It's going to help maintain turgor pressure for a cell. So it's a place for a cell to support and store some of their extra water. The other major part that they're going to have are going to be the chloroplasts. Remember, the chloroplasts are responsible for carrying out photosynthesis. So inside of the chloroplast, you're going to have these little individual discs of thylakoids, and then a whole stack of these thylakoids are called a granum. The thylakoids are going to be where you're going to be doing the light reactions of photosynthesis. The interior here is going to be where you're doing the Calvin cycle reactions in the stroma of the chloroplast. Now, there are several other types of eukaryotic cells, and these are the fungi. They are the protists. Okay, we're not really going to be talking about them a whole lot, but just keep them in mind. They are also very important types of the eukaryotes. That is, that they have a nucleus and these membrane-bound organelles.